Great.
Excellent. So moving on to the last session of the day. So this is kind of building on a theme from last year around standardization and breath analysis and how to start to understand what is on the breath of heterogeneous control populations, normal populations, and then leading into disease cohorts. So building out a BBC atlas. So we've got sessions tomorrow as well. Really excited to welcome partners from the FDA who are giving 3 talks. And what we'll do is we'll take questions in the Q&A as we go through. But all the speakers will come on at the end and we'll discuss all those questions in a panel type discussion. So really excited to have Victoria, Eric, and Omar talking about the wonderful work that the FDA are doing around standardization and understanding what is on breath. So we'll start with Victoria, who will talk about an aim to standardize breath analysis and developing regulatory sciences tools. Victoria, can you hear us okay?
I can.
Wonderful. Great to see you.
Great to see you.
If you're able to share screen, I'll turn it over to you and let you step through your slides. And as I say, we'll do the Q&A right at the end.
Okay, here we go. Let me know if you can see my slides.
Looks like it's coming up. Yep, perfect. Perfect.
Awesome.
Thanks, Victoria.
Awesome. Thank you. First of all, very, very pleased to be here and to be presenting today. And I am joining from DC. So I think among your presenters, I am probably, and my team are probably the closest to the elections today. And it's very, very quiet at the FDA. So we have no band playing here or anything like that. Before I begin my presentation, I'd like to go through the disclaimer, which we have to, as being a part of the US federal government, we'll have to have this as a part of our presentation, that the views that we are presenting today, the data is being presented, it's has nothing to do with commercial products, and it is a view of us as scientists. The presentation, my collaborators are Dr. Epiheimer and Dr. Doran Carranza. We are going to be talking about confident identification of breath matrix and what regulatory science tools are, and that's what we are aiming to do as a part of our study. We come from the FDA, the Office of Science and Engineering Laboratories, which is an R&D arm for the FDA. We are a total of 198 employees. We have around 140 research projects, and these numbers change a little bit with staff coming and going and research and new programs being initiated. At the moment, we have 20 program areas. We have around 400 peer-reviewed presentations, articles, and other public disclosures per year. Our staff is very much involved with regulatory reviews. They act as consultants for submissions that comes from industry to us. And we do around 3,000 or more reviews per year. We're a part of standards committee, and the work that we do, we hope that eventually this work will end up in a standard. So, to be able to help the community. And more than 70% of our staff has graduate degrees in various areas. And our facility is about 55,000 square feet, which gives us a very nice space to be able to accommodate our staff and conduct research to answer some important questions and address gaps that we find. The Office of Science and Engineering, short OSEL, has a mission to accelerate patient access to innovative, safe, and effective medical device through best in practice regulatory science. Scientists and engineers with a broad diversity of expertise are dedicated daily to promoting innovation to impact life-saving medical devices. We have 20 research programs predominantly focused on addressing major gaps and challenges preventing the development of safe and effective medical devices. And these programs advance regulatory science, the science of developing new tools, standards, and approaches to assess the safety, efficacy, quality, and performance of medical device and radiation imaging products. We are a part of CDRA, Center for Device and Radiological Health. Now, you've heard since I've been giving this presentation, you've heard that I've been talking about regulatory science tools. And so what are they? A regulatory science tool is an innovative science-based approach or methodology to help assess the safety or effectiveness of a medical device or emerging technology. Examples would be phantoms, methods. These could be analytical methods, softwares, model, data, and recommended best practices. So for example, in the birth arena, the way you sample and the way you manage your sample can be a best, if we could come up with a best practice, could actually benefit the arena very much. Tools are well characterized for their applications, broadly available, and have utility beyond any individual device submission. They can be voluntarily used across the whole product lifecycle. Tools are intended to ease the burden on innovators to design and validate their own testing methodologies so they can focus on the development of safe, effective, and innovative medical products. If you look at the total life cycle of a product coming to the market, you start from discovery and ideation into innovation and prototyping, preclinical, clinical, and then regulatory decision. The regulatory science tools are very effective at pre-regulatory decisions. So when you're actually in the process of putting your package together for submission. And we have a number of tools on our tool catalog, which is used very effectively in the area of preclinical and sometimes even in the clinical area. Regulatory science tools and their benefits to the medical device innovators actually has a number of facets. First and foremost, regulatory science tools do not replace the FDA recognized standards, but they actually contribute to either new standards or amending the existing standards. These tools represent peer-reviewed resources for companies to use where standards do not exist yet. Regulatory science tools reduce the need for device developers to design ad hoc test methods and allow them to focus their limited resources on how well their new product works, not how well it may be tested. These tools represent an important contribution to reducing risk in all stages of product development and are particularly important to early innovators and inventors who often don't have the means to evaluate their system. Outstanding chemistry challenges, that would be our domain, can benefit from a tool that furthers the science and adapts towards regulatory submissions. And finally, the catalog of regulatory science tools can be helpful for medical device companies to consider when standards or qualified medical device development tools, MDDTs for short, do not exist. This shows the evolution of a regulatory science tool. So it would give you a very good overview of how a regulatory science tool comes about. You can start with a publication, and that's essentially what our team does. They do the research, they conclude a project with a publication or several publications, and then that publication gets turned into a regulatory science tool, and then it's put on our regulatory science tool catalog, which is open to the public free of charge. You can get onto the catalog and download the tool to use. The regulatory science tool can actually contribute to FDA guidance documents, and finally, it can actually contribute to standards, board, and regulatory acceptance, broad regulatory acceptance. So either you can contribute to a new standard, or you can edit, amend a standard. The regulatory science tool on our catalogs look as such. I've just taken a screenshot so you can see how it looks. The program areas are on the left-hand side. So if you don't want to go through the whole list, you can actually click on the program area. And our program area is materials and chemical characterization. And then you can go into the tool and take a look and download it and use it at your leisure. Now, all of that is great. How does our work relate to the breath analysis? We have started a program here at the FDA where we are looking at the biomarkers in breath, because we believe in patient safety and patient access to new technology. We also believe in doing that in the least burdensome, to have least burdensome regulations while we're maintaining a scientific rigor. And we feel that regulatory science tools in this area can be very beneficial, and hopefully one day to contribute to ISOs. The first and foremost area that we are considering and concentrating on is on TB, and with collaboration with private industry and philanthropists, we would like to provide a reference database of background spectra for healthy breath and positive and negative TB control samples to identify potential biomarkers. So confident identification of biomarkers is going to be very important. We are aiming and we're intending to look at sample collection processes, sample treatment, sample storage, sample preparation for analysis, the analytical method and the validity of the analytical method across labs, instrumentation, transferability of the analytical method to different instruments, and criteria for confident identification of biomarkers and molecular signatures. We currently have 2 projects open, which my colleagues, Dr. Eric Miller and Dr. Omar Rivera, will be presenting on these projects right after my presentation. The first project will be presented by Eric and his team, 2 members of his team who have been at the bench doing the experiments, and they are focusing on confident identification of breath matrix compounds, and the results we intend and we hope that we will have an RST to serve the breath community. The second project, which Omar will be presenting, is evaluating analytical method performance for breath analysis, and that is related to lab performance from one lab to another. And we intend to have an RST that will serve that area. The last slide that I will have is the group that we have at the FDA. Every person that you see one way or another is working on projects, several projects to develop regulatory science tools to close out the gap that we observe. Our ultimate goal is to replace animal studies as much as possible and replace those studies with analytical tools that can provide sufficient and reliable information. The most important people, I should say, and not that the rest are not, but are our postdocs who are at the bottom row, who arrive here every day, and they are at the bench every day collecting data. The 2 postdocs who are contributing to our first project will be presenting today alongside Eric on the results that they have collected for us. The last thing I would like to show is how you can get to the regulatory science tool on the FDA catalog, and I will be more than happy to answer any questions regarding the catalog and the use of the catalog. And finally, we would like to acknowledge Bill and Melinda Gates Foundation for their interest in our work, Alstone for their invitation for us to be here, and our management for their support of us presenting today, and our team that constantly, consistently is delivering on important things for us daily. Thank you.
Thank you again so much for taking the time to run through the presentation. Quick reminder to everyone, post the questions in the Q&A on the right-hand side. So first question here is probably more for Victoria. I think a lot of people that are more involved just on the pure research side of things. I was wondering if you could explain further in layman's terms, what are regulatory sciences tools and why are they important for those different types of users?
Yeah, so that's why I tried very hard in my presentation to cover it, but if you haven't used it before, it could be very difficult in first pass to understand what they are. Regulatory science tools could be, they are simply a, in our area, let's pick an example. In analytical chemistry, they can be a recipe for how to do an experiment and how to analyze it. It could be an analytical method, which would include sample preparation, the analytical method, and how would you set up your system, what would be your parameters, and it would guide you in a way how to do an experiment. It becomes very important when you are looking at an area where maybe the ISO is not covering it. Maybe there is a gap in the standard and you need some understanding how to do a certain experimentation. Therefore, the RSTs are aimed to address those gaps and teach you how to do the experiment and get the data in support of your submission. That's the aim that we have with the RSTs. Ultimately, what we would like to do is to either help close the gaps in the ISOs or actually develop new standards for areas that standards are not available. And these RSTs in combination with the publications can actually do that. The next thing that I think is very important for the folks who are on the call today at this conference, that to know that we take feedback on our regulatory science tools. So if you download a tool and you're using it and you find something that you think it's very important for us to know, please, there is a feedback section on that tool. Provide your feedback because we go in and we check to see how we can improve these tools. So for example, if you ran a method and you got a little bit of a different answer that is concerning, we would like to know that. We also look at the traffic. If a tool is receiving a lot of traffic, that means that we are moving in the right direction. If a tool is not getting a lot of traffic, then that means that maybe we're not doing things right. Maybe we should go back and think why this tool is not receiving the attention it should. So in layman terms, a tool can do a lot of things, and we are hoping to be able to allow for those companies that are maybe younger, maybe they don't have the means of having a huge regulatory group, to use these tools to be able to cut down in the process and help them, with their technology development. That's our goal.
Fantastic, fantastic. Next one, I think probably for you as well, Victoria. Could you comment on an example of a regulatory science tool in the catalog? I know you shared a screenshot and there was a link there, but I was wondering if you could elaborate a little bit more on maybe an example.
So there are a number of examples in our group, the chemical characterization group. Our most recent tool, and I think Billy, you and I have talked about this before, our most recent tool that went on the catalog was May of 2024. It's a tool that has looked at 107 reference standards for materials that goes into medical devices. And we are trying to, our aim is to develop a coverage map for these materials that are used in medical devices. And so this tool is very important because it will tell you, okay, if you are doing a submission, you may want to take a look at this and evaluate your material. The tool is so important that within the first day, the hit on the tool was 4,000 4,000 people actually went in and took a look and downloaded. That tells you how important the area is. And we're actually continuing. The next tool that is related to medical device reference standard material is going to come out. We're starting to work on that, and we are going to cover a number of materials. So one would be additives, the next one will be polymers, oligomers, and monomers. The next one will be, colorants. There are, we take all of that into consideration, and a lot of work went into getting the tool together and put it, because we had to look at a lot of chemicals and consider whether these are actually ending up in a device and then manage it from that. There's a whole team that works on that. They're not here today. Omar was partially a part of that team for a while until he moved into the breath work. But yeah, that's an example of a tool that we have recently put on our catalog. 2 more is in the process of coming. As soon as our publications are approved by the journal, the tool package goes into our tool team and they look and review. It gets reviewed, and once we go through the corrections that they ask us to do, and it's approved, and then it goes on our catalog, and it's available to the public for free to use it. It's open to public. So that's how it works.
That's great. I think it's interesting because you can see how there's read across in chemical characterization. There are 2 similar types of challenges that exist in the breath space. So it's a wonderful foundation for a lot of the challenges that the breath community face. Next question, maybe for Eric, it kind of relates to some of the challenges within the breath field. You mentioned the point that getting chemical standards, it's expensive, it's hard for small groups to do, etc. I was wondering if you could elaborate a little bit further on what you think some of the historical challenges may have been for breath, and how some of these approaches around confirming the ID, ensuring something is on breath, may help in the future.
Absolutely. Great question. Historically, the challenges were, I'd say, sample collection, capturing all these volatiles without background contamination has and continues to be a huge challenge. Instrumentally, we've come a long way. Now we have high-res instruments, many different kinds to help confirm that identification. But historically, I think it's been sample collection and high-resolution mass spec, so quality of data issues. Now, I think with many different manufacturers making great high-resolution mass spectrometers, and then also GC technology is, expanding with, 2D GC with the ability to separate so many volatiles in a sample. I think that's contributing to our ability to identify many of these trace level analytes in the breath. So I think moving forward, we'll see a lot of confident identification coming out in the literature. I think streamlining the workflow will really help labs communicate and verify identifications so that the whole breath community can work together and move forward and then do these type of quantitative analyses.
Yeah, no, that's great. And definitely you can see, as you say, with the different technologies becoming available, if you know what the compounds are because you have a confirmed ID and the concentration, that does help you use all of these different analytical platforms and then have comparability across data as well, which I think is going to be fantastically exciting for the field. Next question here is probably for Omar. It's kind of two related ones. So except Python, are there any other tools such as AI that could be used to predict retention?
Yeah, absolutely. There's plenty of more sort of particular tools used to predict the retention in general. We've explored several different techniques in general when we were sort of in the initial stages of this particular study. So in some of the studies that we've actually have modeled, some of our semi-empirical approaches do take an AI and in essence also sort of like a machine learning type of techniques in general. We're not using all of those very complicated particular type of modeling, because one of the pitfalls that you may fall into is that you have to generate an insurmountable amount of data in order for those particular models, in essence, to sort of work properly. So on our end, that's why we sort of have taken a sort of a semi-empirical approach, and we're not using any sort of complicated, you could say quantum chemistry sort of programming, just because in essence, we're sort of trying to test all these particular methods without generating a high level of or generating a high level of data. So yeah, there's a lot of techniques out there. One of the ones that are that we also kind of try to model a lot of our things were from neural networks in general. So there's plenty of techniques out there that you can definitely model all of this based out of. It's actually what you use, what works better for your actual type of application.
Yeah. Next kind of follow on to that, and this is one I was thinking as well. Is there effort to include other mass spec methods other than GCMS, specifically without GC? So I know a lot of the breath research community will use SIFT-MS or PTR-MS. Do you think there's kind of scope to use these same types of techniques for those other analytical instruments?
I don't know if that's directed at me, but at least
That's at you. I would imagine that one.
So I would not for a particular project that we're doing currently for breath. It's another project that we're doing here internally. We're not just focusing completely on using GC techniques. We're focusing on LC techniques. But as far as what I'm currently doing, I haven't explored any sort of other techniques that sort of don't employ GC. So that's something that definitely I could discuss within my actual work group to see if there's any other sort of techniques we can implement to this type of research. I'm pretty sure there's plenty of other mass spec techniques that we can apply to what we're currently doing.
Fantastic. Next one, I think, is probably for Eric. So you talked about the low res and the high res instrument in the talk, and you outlined our things like GC-GC. Is there a plan to do GC-GC, or will it be using different column chemistries in your analytical work, Eric?
Our short-term goals is to use different column chemistries and to continue our current work looking at different stationary phases. Yes, but GC-GC is not off the table. In the short term, we will have 1D GC, high resolution. But I do foresee, because it's a very popular technique in the breath area, I do see tandem GC in the future. However, I cannot say how soon that will be. But we have an excellent lab here, and we're always expanding our instrumentation. So it's something we've been thinking about.
Excellent. Thank you. And again, I suppose if different groups are IDing and quantitating, then we can coalesce different data sets together as a whole field. The next question is for you as well, Eric. Is NIST library the only reference library being used currently?
Great question. No, we're not just using NIST. We're also using Wiley, Agilent, and other resources out there. We prefer to use publicly available databases to more accurately pull from readily available sources and not favor any commercial databases. So we're looking at as many sources as we possibly can. NIST is the most used, and so we thought that would be the best place to start.
Fantastic. Next one, this is for Omar. Fantastic talks for Omar. Do you think a similar modeling approach could be used for prediction of pre-analytical sampling efficiency, e.g. for prediction of recoveries of on-breath VOCs using different sorbent types?
Oh, that's a fantastic question in general. I was seeing it here in the QA. You know, we really haven't discovered that particular part of a pre-analytical sampling efficiency. That would be also one of those particular topics that we could look more into in general. That's where, and for this particular talks, that's why I take some info in general. So that's one of the topics that we can also sort of dig into for future projects. So as far as in general for different sorbent types, I don't know if the person that asked the question means different sort of columns that we're using for this type of work. If it's more related with the columns, then yes, at least for my ongoing work or future work, I would say within the next couple of weeks, I'm actually going to be trying all of these experiments that we've designed in the laboratory, and we're going to be applying pretty much everything to different types of columns. Analytical columns. So some of the reasons being is that a lot of our modeling also takes into account the packing inside of the column in general, their stationary phase. So we're sort of designing all of these experiments in the laboratory so it could well align as well with a lot of our computational modeling. So hopefully that's what the question was referring to, more like the column packing, hopefully.
Thanks, Omar. Time for two very quick ones. This is for you, I'd say, Victoria, and this is something that we've talked about before as well. VOCs, not just in breath, but if you look in the head space of other samples, like a blood sample or urine, you would expect that, you know, that's fundamentally where the VOCs are coming from. Is there plans to look at other sources of VOCs from these other sample types?
Yes, and there are a few things I'm going to cover for plans in the future. Obviously, what Eric and Omar and the team have presented is just the beginning of our work, Billy. I think, and we're focusing predominantly on GCMS because that's where we think that the start has been to develop for breath. You know, the studies are certainly whatever is in literature and what we have found. And if we are going to build the base, you know, you should start where there is some data so that you can take a look. And predominantly, I think GCMS has been something that they have looked, whether it's Orbitrap or something else. Plan for other methods, possibly. You know, I think once we get our handle on this confident identification using GCMS, then if there is a potential for other methods, then we can address that. That would be probably the next level, I would say. I think it's important for us to build the base and then kind of move over. Plan for other areas. Yes, other body fluids are very important because I think it will simply add to the confidence of the breath. If you could correlate, if you could do the work and then correlate the biomarkers that you see in breath with other body fluids, I think that would be tremendous because then it would add to the you know, evidence that one needs for the work. And we have plans to do that. And we are thinking about it. We've just started this work the group has been moving tremendous. I mean, these guys are every day they're doing something. And we started this in April. And since April, we've come, you know, quite a bit in an area that I have to tell you, none of us were an expert. And just by studying and working and talking and learning, we've brought ourselves to this level. And of course, we are analytical chemists by background, but we had some, you know, steep learning. We are hoping to use TB as a base because it's a disease that possibly it's easier to look at, and there's a lot of literature out there that one can rely on, and hopefully we can cross-check this biomarker to get this confident identification if we're all seeing the same thing. And then we would like, if possible, for us to move to the next and the next and the next. So that's our plan. And that's what we're aiming for and hoping that we're intending in the future to expand to other body fluids and expand into other diseases as well.
Fantastic, very exciting. And you mentioned earlier that, you know, always wanting feedback. I love this last question of the session. It's how can members of the breath community help in this effort?
A lot. They can do a lot. So, you know, we're always open to working together if there is a possibility. If there is a situation where, you know, someone is using another piece of instrumentation that we don't know about, it would be nice to know about. I think collaborating with the breath community, all of us together can bring that confidence that we need in this area, in this arena. And feedback, however you can add value to this and share, I think it's going to make us better at what we're doing.
Wonderful. And on that note, I want to say a final thank you for all the contributions today. It's been really exciting, and we're really appreciative of you joining us today.
Thank you. Thank you. It's a pleasure. Thank you for having us.